Good morning, everybody. Good morning or good afternoon, depending from where you are watching this presentation. I welcome you in behalf of CHROMA Technology and all CHROMA Technology employee owners. The presentation I'm going to give is not a product presentation as this was recommended. I didn't want to give you another talk about interference filters and how they are better to use compared with absorption glass or colored glass. What I prepared for the first Photonics Plus and have a chance to give this talk right now is to give you a short overview on what the pandemic situation from the last 15 months influence the photonics market and what we can profit from and what the world can profit from the photonics helping to fight pandemic situation. So we are facing more than 1 year now of the pandemic situation, and whatever we take with us, it's very clear that the world has changed, and whatever comes next is a new normal and not the same thing and situation we used to live in 2019. And a very important tool, as it turned out, during the first month to evaluate the infection and spreading the virus around the world is real-time PCR. While before 2020, only specialists in biochemistry have an understanding what PCR means. These days, everybody knows PCR is the tool to measure virus infection in certain populations. The vaccine development was the main and global task, and I think everybody was pleased to see that end of 2020, the first vaccines were released by the national health organizations, and there's a big hope that end of summer the majority of the people worldwide are fully vaccinated. Having this said, the main thing to get over the pandemic situation was clearly to produce the vaccine in a very quick and timely fashion, and also have the logistic behind it that the vaccine get into the arms of people worldwide. The last question is, what is next? How can we be prepared, and what do we learn out of the pandemic situation? I will go on this later on. First of all, I want to give you a short, deeper introduction into real-time PCR. So PCR stands for polymerase chain reaction, and this is a biochemistry reaction that is amplifying, in this case, the virus RNA up to a detection level. Technology this was invented in the early 1980s and has established in labs worldwide in the 1990s. With the late 1990s, the real-time PCR was established, which enables the lab technician to get a very easy quantification of the genome that was amplified by the PCR, and the quantification is done by a fluorescence response. So these days we are talking of a very rapid testing if there's a simplified preparation, and you can have results within 15 minutes. The whole diagnostic is done with a detection of a fluorescence signal of a family of different fluorescence dyes or fluorochromes, The most used are FAM, HAX, ROX, or Sci5. For the excitation, in these days with state-of-the-art machines, LEDs are used, either white or color-specific or a multi-band LED that covers 1, 2, or 3 of the fluorochromes mentioned in the first line. What is needed from this optical standpoint is a set of filters. First of all, a cleanup filter to just catch the light for the excitation of the fluorophore, and this should compensate a half cone. We all know that the LED beam is not well collimated, and interference filter design are able to compensate half cone to a certain degree. The signal will be catched by a photodiode and needs an emission filter that also cuts off the LED excitation light. And depending on setup, sometimes beam splitting, dichroics, or multi-band filters are needed. Besides the real-time PCR, other optical instruments are very helpful for virus detection and pandemic research. So just to give you 3 examples, I have here plate reader and high content screening systems. They are used for antibody testing, which is very important for the vaccine development, and also for the vaccine production, but also to identify and diagnostic of clinical studies. What is also a very important tool in fighting virus pandemic is the sequencing technology. If it comes to identify the virus genome to develop the vaccine, but also to monitor upcoming mutations, a thing that we are facing right now with the spreading Delta mutation of the Covid-19 virus. And at least, and again, also helping to with clinical studies to get a good overview on the results. And as an outlook for the future, this technology is clearly also a key in pandemic prevention if we think about how can we be better prepared for upcoming situations like we faced early 2020. So what is needed for the different technologies, plate reader, high content screening, and sequencers? Clearly lasers or LEDs for excitation. In many of the high content screening systems, confocal scanners are built in, and on the detection side, high resolution cameras are used to enable the researchers to have a subcellular result of infected cells. This all requires a lot of different optical components, and with thinking of filters, multi-band filters are required. I'm coming to the end, so I have to speed up a bit. More photonics that can be helpful in virus research and fight is Raman technology for production monitoring, especially in the pharmaceutical routine for vaccine production. Microscopy for diagnostic and basic research, and POCT, on-site diagnostic, new technologies to easy detect virus on a cell phone base, like the CRISPR technology. The market potential is huge. We see that the real-time PCR is going smart. There are more and more handheld and on-site compact systems with an easy sample preparation that can be used on airports or exhibition centers and big hotels, shopping malls, and so on. For the year 2020, we saw a 500% increase of pandemic-related optical components. This is consolidating right now, but in some areas of the world, we still have an ongoing increase in business related to the pandemic fight. And this is not for the COVID-19 virus specific. This is easily adaptable to all different types of infectious material like bacteria, other virus, and so on. You also can think about one cell organism like malaria. I'm coming to my last slide. Just a few words about CHROMA. CHROMA was founded and is headquartered in 1991 in Vermont, USA. This is one of the New England states in the Northeast. CHROMA is employee-owned. This year, we are reaching a number of 160 owners. Our filter production is 100% in the USA. We have sales offices worldwide in Europe, Asia, and on the west coast of the United States. Our capability goes from 190 nanometer in the UV up to 3.5 micrometers in the NIR. And CHROMA is known to be focused on custom production according our customers' needs. And we also are known that we are flexible in terms of small custom batches to help our customers with easy prototyping and then go into serial production. Our technology is state-of-the-art hard coating. We have different technologies in our facility and have in 2018 doubled our space and about 30 coating machines. So this is the end of the presentation. I'm going to thank you.